For us, we are using a native promoter for the gene to regulate the expression of SSADH. The rationale behind it is that, as I said, because the gene is expressing in a different level in different cell types. So how are we going to do that? How are we going to target that? Right. So nature provides the best answer because each gene, they are regulated by their endogenous regulatory elements, which there are so many studies already talked about promoter studies and regulatory elements and dysregulatory elements and so on. And we know very well about that every gene, they have a certain regulatory element to promote or to inhibit the expression. And that's why there is promoter concepts that usually is found, this promoter region or regulatory elements are found upstream of transcriptional starts. So that's where we started it, using a native promoter and try to mimic or resemble the expression pattern of the endogenous gene. So this is something that we are heavily invested and using this strategy to make also our vector. And the question is whether it is true or not. So this speaks to the biodistribution, and this is something that we need to test it empirically. And that will be, I would say, it's something that we are focusing on right now, is to see once you inject this virus, which you have incorporated this regulatory element, that we hope that is resembling the endogenous pattern. Is it true, right? So using a biodistribution approach, looking at different different organs, and looking at the protein expression, different organs across the animal, and particularly because we are targeting the brain. So then we can do in situ hybridization, for example, looking at the cellular level at these different cell types. Do we see a transgene expression in those cell type that is relevant? But in those cell types that might not express this transgene, this gene endogenously, we might see the transgene also has a very low level. So that pattern needs to be figured out, and that's where we are studying, and that could combine with some of the capsid that we are using right now. So we are using a tool capsid, a PHP.EB, which is a capsid that has a very high blood-brain barrier penetrant, and we injected the animal by IP, intraperitoneal injection. So the idea is that we systemically, deliver the virus, and just to let the virus go into the brain, because we know the virus capsid has a very good blood-brain barrier penetrant. And then we want to see whether it is once the virus is delivered into the brain, does it really mimic the endogenous pattern? So that is something we are working on heavily, looking at the in situ hybridization, and looking also at different organs, and then to see expression. There are, of course, other regulatory elements or regulatory mechanisms. For example, in our speaker, Dr. Stephen Gray talked about using a microRNA strategy, and those might be relevant for us, and we don't know. We have not tested it, and but that could be something of more like a self-regulatory mechanisms, which we have not tested, but that could be something down the road that we want to study. And there are also some other small promoters that has been characterized that might also be relevant, but in our case, we want to restrict the expression in certain cell types. So a ubiquitously expressed and a very strong promoter for us might not be that useful. It could be a good start, but it might not be the end product that we use in the patients. And yeah, so there are others self-complementary virus that has a very high expression also, and very quick expression once injected into the organism. For us, that is also something that we want to think about. The rate of expression might be relevant for a successful therapy. So as I in the introduction, I talked about that in patients, they have accumulation of GABA, but they also have seizures. And one reason that we found actually also clinically proven is because of the compensatory change of the receptor. Imagine the brain is a very plastic organ and is sensing the changes all the time and is actively coping with this situation. When GABA is chronically high level, the GABA receptor is actually downregulated. So in that case, if we replace the enzyme, then GABA we reduce. But if there's no compensatory upregulation of the receptor, then you will have a problem. So if you have a very abrupt reduction of GABA because of a quick expression of the enzyme, which might not be a good thing. Right? So this is something that we also thinking very heavily, and hopefully the mouse can tell us, because they have a situation where it's mimicking the clinical situation. The receptor is found to be reduced in the mouse. So when we quickly inject the virus, quickly re-express the protein, and then we can ask the question whether this compensatory upregulation of the receptor is it in place? Is it catching up with that downregulation? Is it you're just doing something that might help or might be not helpful or even harmful for the animal? So this is something that we are testing these different parameters of AAV. And the other thing that we're also thinking is the delivery. So right now, I've told you everything seems to be very nice. We injected, IP, and then it magically go to the brain of the mouse. In human, it's a different scenario. The mouse brain is much bigger. The human brain is much bigger than the mouse. So how does the virus, spread inside the human brain? Is so different from, just from a physics point of view. The diffusion of the virus, diffusion of the liquid is going to be so different, and we know very well about that. And that speaks to the route of administration. ROA of gene therapy. That's how we're going to deliver the virus at the end in the patient compared to the mouse. So that's the other thing that we are testing. Given that we know we need to cover the whole brain in our situation, so we think that injecting directly into the brain to the ventricle, so ICV, intracerebral ventricular injection, might be necessary. Basically, we need to bathe the whole brain with the virus, and to see if that will, make a difference in that. So for that, we also need a corresponding capsid for the delivery. So these are the things that it's like a package that we need to think about when translating from preclinical to the clinical side, and that's actually one of the kind of the goal that we wanted to get out from this workshop. Is to understand what is the pipeline, what is the strategy that when thinking about translation from the preclinical side to the clinic, right? From the mouse model to a human, or maybe in between, do we need a non-human primate to mimic that kind of restoration, to mimic that kind of dynamics of delivery?